No, thank you very much for the invitation. It's also a pleasure to work with Nanian and also to present in an international meeting as well. It's very exciting. Okay, well, to start with, I come from a laboratory in Göttingen in Germany, in the group of Professor Niels Vogt. And one of the key focuses of our group is to examine the cellular mechanisms of atrial fibrillation and how this disease essentially works. In order to study a disease like atrial fibrillation, we must have access to atrial-specific cellular models to use. And this is quite tricky, as atrial physiology cannot be replicated with typical expression systems. So we usually are restricted to either animal or human atrial tissue. Another area where we are restricted in our electrophysiological characterization is, of course, the typical low throughput nature of manual patch clamp. So today I'm going to introduce you two of our models, essentially, that we use to characterize atrial physiology, and also our recent solutions to increase the throughput of our electrophysiological measurements. So basically, for the majority of this year, we've been working very closely with the Munich Nanian branch to explore the capabilities of the SyncroPatch as a incredibly high throughput solution to this typical problem of manual patch clamp. So I'm going to give a small introduction to this device. Essentially, I know that Marcus has just presented a very nice overview of the chips and the different functional characteristics of what this machine can do. But for those of you who may still be new to this device, I'll give a very, very basic overview, similar to what myself has experienced in coming to get to know this machine. I traveled to Munich several times this year to be able to see some demos and to be able to use this machine with our own cellular material. And that learning experience has been really, really useful. So overall, of course, the SyncroPatch 384 is a high throughput, fully automated patch clamp system, which has been engineered so one can examine 384 cells at one time. A majority of excitable cells can be used with this system, and of course for us, we were particularly interested in its capability to measure cardiomyocyte electrophysiology. Can it be done? So for us, we operate as a traditionally manual patch clamp lab, and sometime earlier in the year, I took a little survey of all of the manual patch clampers within our institute. How many cells can they achieve on a normal, good day using, in this case, human atrial cardiomyocytes? And their consensus was around 5 cells per day to get good, stable recordings. If we do the math, that corresponds to about 1 cell every 6,000 seconds. So not very impressive, lots of big numbers. We extrapolate that to an automated patch clamp device like the SyncroPatch, suddenly that turns into 1 cell every 22 seconds of the day. This increase of over 20,000% means, essentially, that 1 year of manual patch clamp work from a PhD student, such as myself or my colleagues, can get transferred into 1 single day of experiments with the SyncroPatch. So during one of our first visits to Munich and one of our first demos, Marcus Rapidius was extremely useful and kindly gave us a very, very nice detailed overview of the device itself. And I've taken a little video here which will outline the main surface characteristics of the SyncroPatch. Like, what are you looking at when you stand in front of the machine? So if I pause the video here, the first aspect that I'd like to point out is the Nanian chip. So this is the amplifier housing, essentially, where the 384 well plate is inserted. And this is, of course, where all the magic takes place. Secondly, we have a large array of white trays here in the middle, and these are all where the external solutions are kept and manually poured prior to an experiment beginning. Whether they be reference solutions, drugs, these are all stored in this central array here. Thirdly, on the right, we have the cell hotel. This is where cells are manually inputted early in the experiment. This contains the capacity to keep cells heated at a experimenter-independent temperature, depending on what they would like to do, and also can oscillate to keep the cells nicely in suspension and prevent aggregate formation. Lastly, we have this device here, of course, the robotic biomech liquid handler, incredibly important with its 384 pipette or pipette heads, I should say, in terms of first transferring cells into the nanoion chip and then eventually either applying external solutions, washing, or adding drugs. So also a very basic overview of how automated patch clamp essentially differs. And of course, I'm sure everyone here is familiar. Instead of manually approaching with a very small glass pipette attached to the amplifier, as is the case in traditional patch clamp, we now have a fully automated setup where the cells are introduced into the well with the biomech liquid handler and through gravity and suction pressure are allowed to settle to the bottom of the plate and eventually attach to the patch clamp hole. So a little more of an introduction. This is again more for the people who are not so familiar with the 384 well plate. Of course, we have this very nice plate here, which Marcus has been doing extreme updates to over the last year and a half, as you just heard. And within this plate, cells are pipetted with the biomech robot from the cell hotel into the chip itself. And during our demonstration, a little video here where the cells are now inside the pipettes of the liquid robot, and they are automatically pipetted into the nanoion chip on the left-hand side. So it was very interesting for us as primarily manual patch clampers to essentially learn about this technology and to see, is it actually true that it is essentially as simple as that? Can a cell just be pipetted into this 384 well plate and eventually settle on the patch clamp hole? So sometime substantially after an experiment involving iPS-derived cardiomyocytes, we took the plate and just popped it under a normal light microscope just to see if we really could see the remains of a cell still over that patch clamp hole. And if I play this video here, once I adjust the focus on the microscope, you can see the remains of, not very pretty remains, but still the remains of a cell directly over the patch clamp aperture there. And for us, that was quite exciting because that's really the picture out of the textbooks in a way, where the cell perfectly, once at a time where it was healthy, was sitting perfectly on that patch clamp hole and would have provided a measurement during the experiment. Of course, the software user interface has been thoroughly engineered by Nanoion, which is really, in my opinion, very nice and to visualize exactly what's going on. And here was a typical experiment where the green squares here indicated a successful gigaseal within our test of iPS cells. So, speaking of iPS cells, I'd now like to get on to our first atrial model that we utilize in our lab. This is involving atrial-specific induced pluripotent stem cell-derived cardiomyocytes. We use standard protocols with mesodermal induction using shear and then pushing towards a cardiac phenotype using IWP2. And the unique part of our protocol here is that early in differentiation around day 3, we add a vitamin A derivative called retinoic acid into the differentiation medium. That's highlighted here in the red box at day 3 there. And what this does is this can push the cells to generate a more atrial-specific phenotype during their differentiation process. So at the end of their typically, let's say, 25-day differentiation process, we can then generate reasonably pure populations of atrial-specific cardiomyocytes. What this looks like in culture is if we stain for either ventricular-specific markers, such as the myosin light chain ventricular isoform, which is here shown in green, and in contrast, the atrial-specific isoform of myosin light chain shown in red, we can see that in typical differentiations of untreated cells, we see a large amount of green stain, therefore a large amount of ventricular cells. If we add retinoic acid into the culture and stain for the atrial isoform of myosin light chain, we can therefore see that a large component of the cells now display atrial-specific markers. This is roughly 95% pure, or 95% positive, I should say, for the atrial isoform of myosin light chain. During initial studies of the electrophysiological phenotype of these cells, we were also very pleased to see that they do in fact display typical atrial-specific phenotypes. In this case, a much more shortened action potential with a more triangular shape, more typical for atrial cardiomyocytes compared to ventricular. So as a result, we are quite confident and able to label these populations of cells as either ventricular cells for the cells that do not receive retinoic acid treatment, or atrial cells as the populations that do. So for our first test with Nanion, we took these cells to Munich, and we wanted to see how well the SyncroPatch could determine the differences, the sort of, let's say, small differences in electrophysiology between these two cellular subtypes. Can the SyncroPatch in fact determine with high sensitivity, the differences in the electrical phenotype? So firstly, we used a chip that was split in half for ventricular, half for atrial cells. We had quite a nice cell number. We had 10,000 cells per 10 microliters of medium. We used a high resistance chip and no perforator for these experiments. And firstly, to measure the L-type calcium current, we were able to clearly see subtype-specific differences within our cell lines. The atrial iPS cell-derived cardiomyocytes showed a smaller L-type calcium current, which is very typical of human and animal atrial cells. And we were also very, very pleased. After 1 day's worth of work, we were able to generate a huge amount of cellular recordings from these cells that we just took in our package down to Munich, with 250 ventricular recordings and over 100 atrial recordings as well. For us, sitting in the lab doing manual patch clamp, that was very, very impressive to be able to see these numbers. What we then did is we actually continued the experiment. So immediately after the L-type calcium current acquisition, we changed the internal fluid now to contain a potassium fluoride composition, which allowed for, or I should say unblocked, potassium currents, and therefore we could examine action potentials once we switched the amplifier configuration to current clamp. And again, using the same cells still attached to the Nanion chip, we were also able to replicate typical action potential morphology differences between our ventricular and atrial subtype-specific iPS cell-derived cardiomyocyte lines. So again, we were very happy with this result from the SyncroPatch. This concept of solution exchange during an experiment was also very exciting for us, because of course with manual patch clamp, it is extremely difficult to exchange the internal pipette solution with the intent to measure different currents or action potentials. However, with automated patch clamp, that is not so much of a problem to exchange these kind of solutions. And so simply after changing the internal solution, we were able to measure both L-type calcium current and action potential within the same cells, which was really interesting in a sense to be able to determine the relationship between these two parameters. What happens to the action potential specifically with an increase in L-type calcium current. In a sub population of cells here, we were able to show that in almost 200 cells, a really nice relationship between the action potential duration and L-type calcium current. So now moving on to our secondary model of atrial physiology. Of course, this involves animal cardiomyocyte atrial or ventricular biopsies. Typically in our institute, we are lucky to have access to human tissue as well. And so in the future, we hope to be able to incorporate human heart tissue biopsies and replicate these recordings I'm about to show you. For these test recordings, we used swine hearts. We isolated cells from both the ventricular and the atrium and put them into the SyncroPatch with the same protocols as we used for the IPS cells. Here's a small overview here, a suspended cardiomyocyte. And you can see, got a nicely isolated cell. You can see striations there. And then a similar, not so nice photo after the experiment. We can still see the remains of a cardiomyocyte still attached to that patch aperture there. For these experiments, we had substantially less cells available than we did for our iPS cell-derived cardiomyocytes. We had between 3 and 2,000 cells per 10 microliters. In the end, we used a low-resistance chip, and a perforator was necessary in order to gain proper access to the cytoplasm. Because of our low cellular population, we only used partial chips for both cell types. And here I'm very happy to show you what we believe is the first ever recordings from native cardiomyocytes with the SyncroPatch. Really nice subtype-specific L-type calcium currents here. Again, we see a much larger density in ventricular cells compared to atrial. And simply that enough was incredibly exciting for us to be able to visualize this. In a similar fashion, we then changed the internal solution to unblock potassium channels, changed the amplifier configuration on the SyncroPatch, and assessed action potentials in the same attached cells. And here again, we were very happy to be able to see really beautiful subtype-specific action potential morphology between the two cell types. Much longer plateau, a much more typical ventricular action potential in our ventricular cells, and shorter, more triangular shape in the atrial cells. We were then very interested to push our luck, as it were, and continue measuring from these same cells within the same experiment. And what we did here is we changed the external solution to add a high external potassium concentration up to 20 millimolar. This is in order to shift the reversal potential to more negative ways in order to quantify the basal inward rectifier current IK1. So using a ramp protocol, we then assessed IK1 within these ventricular and atrial cells. And again, we were able to see very typical subtype-specific characteristics. We see a high IK1 density in ventricular cells and a much lower density in atrial cells. And this is exactly what we expect to see, for example, in a typical human ventricular and atrial measurement. What we then did is added an external drug in order to assess an atrial-specific current known as the acetylcholine activated inward rectifier current. We applied carbachol, which is an M2 receptor agonist. And as you can see, this increased basal current, shown in blue, in only the atrial population. The current was not increased in the ventricular cells. And this was also very exciting for us because this method of determining atrial specificity and being able to show that a certain current is only present in atrial cells is, of course, very relevant for our work with atrial fibrillation. So for the SyncroPatch to be able to show this as well was incredibly exciting and really nice for us to see. So, in summary, because this was a first test, really a preliminary test, and I would like to give an overview of our success rates, essentially, for comparing the iPS cell measurements with the native measurements as well. Successful seal application was around 30% in the native cells at first. We were using fluoride-based solutions with an older SyncroPatch model. This was done early this year, these measurements. And we achieved a seal success rate of about 60% in the iPS cells. Moving on is a success rate measurement per current. We had around 10 to 15% success rate of all the cells we measured, meaning of all the cells we put into the device, around 10 to 15% of those cells gave us robust current measurements. So because this was a preliminary test, these numbers, of course, can be improved. And we're very happy to say that after these extensive testing and extensive help from Nanion as well, we have purchased a SyncroPatch device for our institute, which will arrive hopefully in the next few months. And so we're incredibly excited to be able to continue these measurements into the future and see how much success we can achieve using human atrial biopsies as well. So with that, I'd like to say to our kind Japanese hosts, go seichou arigatou gozaimashita. Thank you very much for your attention. I'd like to thank primarily the team at Nanion for the extensive help that they've given us over the last year, essentially, in particular, Marcus Rapedius. Marcus, you were absolutely crucial to the success of these experiments, so thank you very much. And of course, thank you to my team for our stem cell unit here in Göttingen for providing the iPS cells, and for the team at Munich University as well for providing the pig tissue. So thank you very much, and I'll be happy to answer your questions.
Thank you for your presentation, Wilson. Maybe you have a question, Wilson? No? OK。どなたかご質問ある方いらっしゃいますでしょうか。Wilson, I have one question. I think right after dissociation, when you use native cells, the sample should be very dirty. So I think it's very difficult to measure the cells on automated patch clamp because we cannot select the cells. Mm-hmm. Did you do something special to capture the cardiomyocytes from your sample?
We use a isolation protocol that has been developed by our lab over the last 10 years, essentially, or more actually. It's a very good and standard protocol, and that involves multiple filtration steps in order to filter out the debris, as it were, from a typical isolation.
Okay.
And so that would be, I would imagine, yeah, the key aspect into removing debris from the sample.
Okay, so you have very nice skills to remove debris.
We do, yes. I must say this wasn't done by me. We have a postdoc student, Funcho Fakuada, who is incredibly experienced with the isolation procedures. He does it almost every day with human samples, and he was responsible for the isolation of the swine cardiomyocytes that I've shown today.